DNA fingerprinting is a bit complicated topic. There are some terminologies. If we understand those terminologies, understanding DNA fingerprinting will be very easy. First thing is repetitive DNA. Repetitive DNA is that part of the DNA which is having a repeated basis. That is called repetitive DNA. There is something called as satellite DNA. Satellite DNA is a class of repetitive DNA. It is called as satellite DNA because it has got some hidden information. If we analyze that hidden information, we can get some valuable information regarding the DNA or of an individual. So, based on various factors like length and type of basis, satellite DNA is again classified into two different types. One is microsatellite and mini satellite. So there is something called as VNTRs, variable number of tandem repeats. It is nothing but the repetitive DNA which has repeated many number of times. This is called as VNTRs. What it is? It is a repetitive DNA which has repeated many number of times within the chromosome, and repeated units vary. So in this satellite DNA or in this repetitive DNA or in this VNTRs, they have a special feature called as polymorphism. What is polymorphism? A small variation at the genetic level. That variation is caused mainly because of small mutations. Mutations in a repetitive DNA will keep accumulating within the generations, which means repetitive DNA is a non-coding DNA. It will not code for any protein. So if any mutation takes place within the repetitive DNA, it keeps accumulating within the generations. So it becomes easy for us to analyze repetitive DNA and determine the relationship between the two individuals. That is mainly because of polymorphism.